Welcome. In this tutorial, I will introduce you to Fusion 1.0. What Fusion 1.0 is, why it matters, what problem it solves, and a brief overview of how it works. Okay? Fusion 1.0 is a powerful web-based platform designed to bring together tissue structures and molecular data. Okay, it integrates histology whole slide images with spatial transcriptomics data, allowing researchers to analyze both tissue morphology and molecular composition all in one platform. Okay, and Fusion 1.0 currently focuses on kidney whole slide images and VGM data. But why do we need this? Why we need to combine histology image with spatial transcriptomics? The answer is accuracy and more insight. Okay. Traditionally, doctors predict diseases from symptoms. For example, frequent urination, weakness, increased thirst, hunger might suggest diabetes, but these same symptoms can also appear in urinary bladder cancer. So, symptoms alone aren't always precise, they only help narrow down possibilities. The second approach would be tissue analysis, where doctors take biopsy and examine tissue structures and compare the morphology with healthy samples. Okay, while this provides more information than symptoms, analyzing tissue morphology by itself still gives limited insight, and also it's qualitative, meaning it can change from doctor to doctor. Therefore, the third approach and much accurate approach would be to combine molecular data with quantitative tissue features. Okay, and Fusion allows us to do exactly that. It allows researchers to investigate molecular data and quantitative tissue features within desired functional tissue units, which are the meaningful biological compartments, okay, like glomerulus or tubules or something like that. And, and it provides more precise, comprehensive understanding of tissue function and disease. Now that we have discussed what Fusion is and why it's important, let's look at how Fusion actually works. Okay, so the Fusion workflow can be broken down into three simpler steps. First one is upload data, where user gets 20 GB of personal storage for uploading and storing their own data, which can be kidney whole slide image, 10x VGM data, or any other datasets. They can also access publicly available data stored on Fusion platform. The second step is a process where they can use the built-in tools, which we call it plugins, to perform various tasks like segmenting functional tissue units, which can be glomerulus, tubules, or any other biologically meaningful compartments. They can also extract molecular and morphological features, aggregate them, etc. And the third and the final step is to visualize and analyze those integrated data, and they can do directly on top of histology, or they can do different sort of plottings to find out the correlations and biological meaningful insights. So now that you have seen a high-level workflow of Fusion, which includes upload, process, and visualize, let's break it down into a clear step-by-step pipeline, starting from upload to doing integrated analysis. Okay, so the step starts with uploading data, which can be a whole slide image, or it can be VGM data. This is a sample of RDS file and how it looks. So once we upload the data, it will look something like this. there will be one histology image and its corresponding RDS file or any other VGM data, okay, which can be H5 or H5AD, and we are currently implementing FastQ as well. So once we upload the data, the next section is process, which includes these steps. So the first step is metadata injection, which is optional, where users can add context to the uploaded data. For example, this biopsy, this slide is taken from a male patient who was 30 to 50 years old and they had CKD. Like this, we can add more metadata and context to the image. And the second step is functional tissue unit segmentation. So FTUs are biologically meaningful compartments within the whole slide image, which can be glomerulus, tubules, or any other meaningful structures. So this is an example of glomerulus segmentation done using one of our plugins, and this is another example of FTU segmentation using multi-compartment segmentation, where it segments 6 different compartments like glomerulus, tubules, arteries, interstitiums, etc. And after FTUs are segmented, the next step is pathomic feature extraction, where we extract 72 different quantitative features based on morphology and intensity from each FTUs, and then we do spot data generation, okay, where we extract the cell type information, their proportion and states from the VGM data per spot, okay. So once we have this molecular data from the spots, we do spatial aggregation, where we take the molecular data from the underlying spots and aggregate them at a FTU level, okay. So now those molecular data from spots are aggregated into this glome, and we can see those cell types, proportions, states for these glomes, as well as those 72 morphometric features that we extracted. And lastly, we can do visualization and analysis, okay, and these are examples of the visualization. This is, for example, we are visualizing these glomes and we are looking at this spot. So, and from this we can do a hypothesis generation. And so that's pretty much it. This is the workflow of Fusion, and in next video I will do hands-on and we will learn how to use Fusion, okay.